Sweet. All right. Good afternoon, everybody. Thanks for getting your name tags out. Thanks for being here on time. I am thrilled today that we are starting our unit on venom with Dr. Andrew Mason, who you all have met a couple times this semester so far. I won't give you details about him because we've introduced him before, but he'll be leading class the next couple weeks. And yeah, we're really excited about it. So I'll just hand it over to you,
Sure.
And then we can get started, and I will just cover it.
Great. So first and foremost, before we get into awesome, cool things about venom, I do want to wish everyone a very happy Valentine's Day. So these are lizard pelvises. And if you didn't know, lizard pelvises have a nice little heart in the middle. So, I mean, that's beautiful. Herpetology and Valentine's Day, it's all together. I also want to quickly say, it was funny earlier, Princeton asked me something. He said, so are you like excited about teaching? And I was like, yeah, I am. I guess I am. So I don't actually teach a lot in the position that I'm in. I guess I should stay here closer to the mic. And I really like teaching. So I'm excited about getting to teach when I haven't for a little while. And then I get to talk about venom, which I've spent 6 years studying, which is just like, that's it. I'm super excited about it. So hopefully I can share some of my excitement with you guys. And you can take away a little bit about some cool things about venom. So today's agenda, we've got a kind of a, we're going to go back and forth a little bit. We're going to start off and do a little bit of a lecture series or like a short lecture. And then after that, we're going to move into discussing your primary lit review, which hopefully everyone has taken a look at. And you guys are going to give each other some feedback. And then you'll have an opportunity to resubmit those. And then Dr. Yangloff and I will look at them for next week. After that, we're going to come back to venom, talk about a little different part about venom, specifically the diversity of venomous herps. And then finally, we've got another activity that we'll get to at the very end, which I'm really excited about. So I'm going to start off with a question for you all. What is venom? I would like 3 definitions, please. And anyone can volunteer and just give me the best guess you have about how you would describe venom. Yes, Ethan.
Some sort of specialized toxin or saliva that attacks an animal in some way.
Okay, specialized toxin or saliva that attacks an animal in some way. I like it. It's good. Injected. Okay. Let me hear another. Doesn't have to be totally different. I just want another set of wording. Chase.
A toxic substance that comes out of a herp.
Toxic substance that comes out of a herp. Okay, not bad. I need one more. Yes, Sydney. Injected. All right, so we've got a lot of commonalities among these definitions, and that's what I like. I wanted to get those commonalities so we all get a sense. We have toxic. I heard that 3 times. Perfect. I heard injected at least, was it twice or 3 times? It's pretty darn good. And that's in a lot of ways the key components of the definition that we're going to use. So I'm going to broaden it just a little bit. And I'm not going to say injected. We're going to say traumatically introduced. Now, the reason we're saying traumatically introduced and not injected is that there are some exceptions I'm sure you can think of. Anyone ever seen a spitting cobra? Yeah, so that's not injected, but it is quite traumatic if it is introduced to you. It is a natural toxin, and we typically associate venoms with a use for prey capture or defense. So when we say traumatically introduced, again, one of the principal ways that we see this introduced in herps is with fangs. So you can see this is a lovely, I think it's a white-lipped tree viper, but it's doing a great job of showing you these huge fangs, these nice hollow tubes that are going to be connected to the venom gland, and they're going to deliver that toxin with injection. But there are other ways you can, like I said, spitting cobras spit their venom, and if it comes into contact with a mucous membrane, they typically go for the eyes. That is a traumatic introduction. Venom is not necessarily unique to herps. So in fishes, there are venomous fishes that have barbs. These guys have barbs or spines. Those can be another means of injecting venom or introducing it. And then last, we'll mention stingers. So if you're a scorpion, bee, those sorts of things, all use venom. Could be for prey capture or defense, all injected traumatically. So that's our working definition for venom for this class. If you have to remember one thing, hopefully that's what you take away. But we want to differentiate between venoms and poisons, which are the other big thing in herps. And I'm sad to say that I'm actually not going to talk about poisons very much, if at all, even though they are really cool. And we see them in frogs. So this is a quick slide. Quick, if you need to tell if it's poisonous or venomous, just think of this. Maybe you've seen it before. If you bite it, if you ingest it, if you bring it into contact with you with your hands or something like that, that is a poisonous organism. It could be frogs, it could be plants, all sorts of poisons. If instead it bites you, it is venomous. So another thing that was specific to our definition is that we mentioned it has a use. In almost every case, venom does not evolve spontaneously. It's been selected for, and it is often used for prey capture. It's a key thing that we're going to talk about. So with that in mind, I have a YouTube video up I want to show you guys about a specific predator using theirs. So I hope this plays sound. Apparently it's playing it on my computer.
Its great speed and extraordinary senses in a remarkable hunting strategy we are only just beginning to understand. Ember rattlesnake. The morning sun has warmed its body, giving it energy, and it starts to move. It's searching for a place where it can conceal itself and wait for prey to come within striking distance. A little chipmunk. It's in no danger yet. The rattlesnake can't move fast enough to chase and catch it. But small mammals tend to use the same paths as they run over the forest floor, and they leave behind a faint trail of scent. The rattlesnake can detect that scent with its tongue. It can also locate the warm-blooded chipmunk if it's nearby, with heat detectors in a pair of pits beneath its eyes. As it moves, it carefully holds its rattle above the ground so it makes no noise. It's chosen the place to wait at the bottom of this tree stump. Its coloration matches the ground so closely it needs no further concealment. Now it's just a matter of time. Seeing a rattlesnake actually catching its prey is a very, very difficult thing to observe. In fact, some scientists have watched rattlesnakes for years without seeing that particular crucial moment. But we have a chance, partly because rattlesnakes are ambush hunters, so we know exactly where to put up our gear, and partly because in that gear we've got the very latest in surveillance equipment. There are remotely controlled cameras and infrared lights on stands. And there are motion detectors that will switch on the cameras if anything moves. So I needn't wait alongside. If anything happens, the cameras will switch on automatically. Later, I check the replay. There's a mouse, just along that log. That obviously came to nothing, but the cameras have started recording again and the snake is moving. He's checking out the trail with his tongue. See, that's exactly where that mouse was running. It's pitch dark, and the mouse clearly has no idea that the snake is there. But the snake is well aware of the mouse, thanks no doubt to those heat-detecting pits. A snake strikes by suddenly straightening the curve in its neck, but at the moment, the mouse is not within range. He's worked out that that is the path along which the mice run. He's getting himself properly adjusted so he can strike when he next gets a chance. Now once again, waking. That's what snakes are so good at. Oh my goodness. That's a dead mouse, all right. Slow down that shot, and you can see that the snake stabs the mouse just once. After 3 convulsive kicks, the mouse is dead. The snake is moving again. He's getting back now to the one that he knows is dead, back there. Where is it? Ah, now it looks as though he's really got it. That's his dinner. And that can last him for 3 weeks, 4 weeks if necessary.
Right. So if you haven't seen that clip before, which hopefully you haven't, that is a pretty unique view of being able to see a snake hunting. It's not common. Snakes eat so infrequently that you, if you happen to see one catch prey, I mean, you're extraordinarily lucky. As they mentioned, you have to basically set up a camera and wait. And oftentimes you'll see the snakes miss. There are actually some great videos of snakes trying to catch mice and mice very nimbly dodging. Yes, Sydney. Nope, sure don't. I sure don't. Yes.
Yawn after striking?
It did.
Is that
Yeah.
common?
Yeah, pretty much. So we're going to talk in a little bit about the morphology of those fangs of those species. And actually, I've got even another video of the snake yawning. But yeah, they do that. They do just basically to stretch out their fangs, to like realign their jaws. It's exactly what it was doing and exactly what it's for. It's not the same as us, like to catch a breath, but it is to just like get things straightened out. So. Okay, no other questions really quick. So one other thing I want to mention that we saw specifically here in that video was a really specific type of behavior. That was called sit and wait predation. So this is a hunting strategy where you basically pick one spot and you're going to wait. Ideally, you're camouflaged or you're difficult for your prey to see, but you're not moving. You're waiting for the prey to come to you, and then you strike opportunistically. So this is a very typical strategy of vipers. The timber rattlesnake is a viper. We're going to talk about vipers in depth today. But it's interesting because one of the things that we sort of see as a characteristic of vipers that employ this strategy is they tend to have a little bit thicker bodies. They tend to be a little kind of like fatter, a little shorter and a little fatter. They might be considered slower moving overall, although that's not always the case, but they are very fast strikers. So they're not going to travel long distances, but when they have to move that little distance to get a mouse, super fast. And this is in contrast to another type of hunting strategy, which is called active foraging, active forage predation. And this is what you might typically think of as being like a hunter. This is an organism that is actively looking for food. It might be tracking food like mice, or in the case of this king cobra, it could be tracking other snakes. There are actually several species that eat exclusively snakes. And so this is a pretty common life history strategy for elapid snakes. So we're going to build a contrast throughout this talk between the vipers and elapids, basically these two important radiations of highly venomous snakes. And this is one of those sort of key characteristics among others. Give you guys a second. So we've covered lots of information about venom. We now kind of have this working definition that we're going to stick with. But throughout all of that, yes, Sydney. Elapid is a family of venomous snake. We're going to cover that in the second half of the talk. But I wanted to introduce these two strategies early on. And I want you guys to remember that each is generally characteristic of a family. Yeah. Yes, Ethan.
Is there a particular strategy that Colubridae follow, or are they kind of a mixed bag?
They are kind of a mixed bag. Colubridae are very diverse. We'll cover Colubridae as well. So we have this definition of venom, but none of that actually helps any of you know what is in venom, because there's this very different concept between knowing what a venom is and knowing, if I was, for instance, to show you a drop of venom, what is in that. And so if you want to know what a drop of venom looks like, here's an example. This is a sterile collection cup, and that is a drop of venom. It's, in this case, this bright yellow liquid. It is not always yellow, but in many cases it is. But one of the things that I think is critically important to know about venom, and this is something that I actually did not appreciate that many people didn't know, is that venom is a mixture of toxins, especially in snakes and lizards and in most species. It's a mixture. It is not one sort of like continuous, it's not one molecule. And in fact, in snakes, we could be looking at 50 to 100 different proteins or enzymes that make up the venom mixture. And so because of that, one of the sort of terms I see used a lot in the literature, because it's kind of catchy, is a complex cocktail of enzymes and organic molecules. And together these work to incapacitate prey. One of the other things that's really important to know about venoms, and I think about, at least in snakes for sure, definitely in lizards for sure, so herps in general, I think that this is true. I think in all venoms, or most all venoms, the toxins are produced by the organisms themselves. And so this differs from some of our poisonous species. So if you're familiar with poison arrow frogs at all, in fact, the poisons that they utilize, they collect from their environment. So they eat these insects, and the insects have a toxic alkaloid that they sequester in their skin, but they're not making it. In venoms, the organisms are making all of them themselves. So we can look at venom as this cool substance, and it's really neat, but that doesn't tell us a lot about what's in it. We want to know what kind of proteins are in it. So another way we might represent venom, and I'm going to show you this because we might come back to this, especially later on in the week, is by its composition. So in this case, what you're looking at here is a pie chart, and the pie chart represents the sum total of venom, and each of these different colors actually represents a type of toxin. And snake venoms specifically have a bunch of different toxins, but they're only from a handful of toxin families. So each of these colors actually represents a toxin family, not a specific toxin. But in most cases, toxin families are determined because they have similar structures and similar functions. So another way we might represent this, another way we might quantify venom or look at venom, we could look individually at each of these toxins. So this is yet another way we might look at the composition of venom. And here, what you would be looking at, this is the same basically information, just displayed differently. Here, each of these bars represents a single toxin. So look how many bars there are. This is all the venom of one species, that many different proteins. Each of these different colors represents a different protein family. And so we've got lots of different proteins from a handful of different families. Why? What is the point? You know, what purpose does this serve? Well, these different enzymes that are in venom carry different functions. This is super important. And so we have a bunch of different functions that enzymes can have, but there are only two very broad functions I want you to kind of remember. The first is chemotoxicity. So these are going to be enzymes that attack blood, basically, or they attack your what's called your coagulation cascade. It's your body's ability to maintain your blood in a like a mixture that is what your body needs. So there are ways that you can disrupt this. You can cause overclotting. So you could have like clotting forming of your blood inside your veins. You can cause things that stop clotting. And so your blood will actually start to leak from your body. Lots of ways that you can affect this. And on this chart, this is from a publication that was a review of venom. And you can see there's all of these different little snake icons. Each of these represents a different type of enzyme. And they're all attacking these different places in this very complex coagulation cascade. So for instance, you might have metalloproteases are a really important component of some venoms, and they're going to attack platelets and things like that, disrupt them so then you can't clot. Now, this is in contrast to another very broad category of toxins or enzymes that are neurotoxins, neurotoxicity. So these are going to be things that affect your neurotransmitter function. So some of these block synapses, some of them will fill receptor proteins at what's called the neuromuscular junction. And functionally, these can cause a number of effects. The most common one is paralysis. So that can have two consequences. The first is it might paralyze your muscles like your legs. So if you're a mouse, you've gotten bitten by a snake, and you've got a neurotoxic venom that is paralyzing your legs, you're not going anywhere, unfortunately. The alternative is that they could affect other more basal functions, like for instance, your respiratory system. Because for instance, in us, our respiration is powered by our diaphragm. If your diaphragm is no longer working, you are going to asphyxiate. And that's exactly what happens. So these are just different strategies that these species employ to take out their prey. And if you have a venom that's a mixture of these strategies, ideally, you might be able to have several of these different effects. Yes?
I saw a video that it's snake venom, like a thing of blood.
Yep.
That, like, clumps together. Is that literally what that chemotoxic venom does?
That is one function of, that's like one example of the way hemotoxic venom can function. Yes, I've seen that exact video. Sometimes I show it actually, but it can be a little grim. Yeah, so it can really mess up your blood. Yes, Chase.
So a couple of slides ago, when
Uh-huh.
like the bar type graph.
Yep.
So is that just like one species of snake's venom? And it was like, okay, so can every single time it like produces venom, it's gonna produce like that exact like
Yep.
picture, or could there be different levels of different ones?
No, by and large, every time it produces venom, it's going to produce that same mixture. We'll talk a little bit about venom production today. Yes, a couple questions over here.
Like you said, it's like
Or hemotoxic. They do sometimes. We're going to talk about that too. Yes, Sydney.
Are different individuals, do they have like different amounts of venom
They absolutely can.
exacerbated, or is it?
Extremely.
Yeah, is it pretty?
I'm so excited you asked because guess what? We're going to talk about it.
Okay.
Yeah. Yeah, absolutely. You guys are asking all the right questions. We're going all in the exact same direction. That's great. So let's going back here. So if you have, I've got to ask some questions just now. What's the point of having all of these different toxins? They have different effects. If you have these toxins occurring in different amounts in your venoms, that actually means that your venoms will have different effects. So for instance, you can imagine a species. It's expressing or producing a lot of a hemotoxic protein in its venom. And if that is injected into an organism, we expect it will have a highly hemotoxic function. And you could put that in contrast to another type of species or individual even that might be producing a lot of neurotoxin. And then that venom will have a neurotoxic function. So this leads to basically variation. This is variation in venom composition, which we presume leads to variation in venom function. And one of the things that's really cool about snake venoms, at least, is that there is a ton of variation in snake venom composition and function. And it occurs across different biological scales. And that's really fascinating. So I'm going to give you one example to start off with. And this, I think, is pretty intuitive. This might be the kind of thing that you expect, because this is species specific. So here I'm looking at a specific group of snakes. These are called palm pit vipers. This is actually the group that I did my dissertation work on, so they are very near and dear to my heart. And here we're looking at 3 specific species of palm pit vipers that all live in Costa Rica. So this is the eyelash viper, the black speckled palm pit viper, and the side striped palm pit viper. Again, these all live in basically the same country, so that they're not quite what's called sympatric. They're not exactly in the same habitats, but they're in. If you, for instance, walked up and down a mountain, you could run into all 3 species. And all 3 of these have very different venoms. So we'll start up here looking at the side striped palm pit viper. You don't need to know exactly what's in its venom, but you can see how these colors are different. And I know, unfortunately, this guy has some different colors used in its pie chart. But if you just compare these 2 pie charts, you can see how much of a difference there is, for instance, in this big pink chunk of the pie. That indicates that this species has a lot of this one specific type of toxin. It's actually metalloprotease. That's what I mentioned earlier. You don't have to know it. But that means it's got a very hemotoxic venom. And we can contrast that with its buddy. So this species in the middle, this is the black speckled palm pit viper. It actually has a venom, this big red chunk of the pie. That's a very specialized neurotoxin. And so this species, in fact, has almost no metalloproteases at all, none at all. And so if you looked at the functional activity of these 2 species, you basically got 2 ends of the spectrum, where one is going to be having a hemotoxic function and the other has neurotoxic function. And then if you go down to this 3rd species, this is the eyelash palm pit viper. It's actually got kind of a mix. It's got some hemotoxic activity. It's got a different type of an enzyme that actually affects what's called like cytotoxicity. So that's going to affect just general tissue damage. And then it actually has a specialized protein that's going to really increase your blood pressure, which is cool. So 3 species, all in the same genus. So these are all pretty closely related, all with very different venoms. And I wanted to start here because in some ways, these species level differences, I think, are a little more intuitive. They make sense. We know lots of different species that are quite different from one another. If you think about birds, there's a lot of different color variation. So maybe it's not so surprising that there is this much variation among species. And especially if we look at these, remember you guys have been talking about reading phylogenies. None of these are actually sister taxa. So for instance, they're not, their closest relatives. They're at different places in the phylogeny. So maybe let's look at another example. We're going to hold on to this guy, this black speckled palm pit viper. And we're going to look at its sister taxa. So this actually was not represented in that tree because it was recently described a few years ago. And these 2 species are sister in the phylogeny, which means they are each other's closest relatives. I'll say this as well. One, they're morphologically very, very similar. It's extremely hard to tell these organisms or these two apart. You basically have to be counting scales because if you're just looking at pattern, couldn't tell them apart. They occupy basically the same habitat, slightly different parts of a mountain range, but they're both high elevation specialists. And yet, these two pie charts are the same. These colors are the same between them. You can see how different they are. These are two very different looking venoms. And we have this same actually contrast where our black speckled palm pit viper, remember it has a neurotoxic venom and its sister has a hemotoxic venom. So this is an example where we have sister taxa that are extremely divergent. They have very different venom compositions and venom functions. And although this is cool, it gets even wilder because we also see within species variation. Yes, Sydney.
Sorry, when you were talking about the visors,
Yep.
do they eat the same thing?
We don't have very good data on that, but probably they're probably similar, but I don't know for certain because we don't have great data on it. Yeah, but that's good thinking. So here's another really famous example of snake venom variation, actually. So this is a Mojave rattlesnake. Has anyone been out west by chance? Sydney has. All right. How far out west? We talking Arizona, New Mexico? Okay, New Mexico should have them. Anyone else? Yeah. Arizona. Okay. Did you guys hear anyone say, depending on where you're at, you might have heard someone say, watch out for Mojave rattlesnakes, watch out for Mojave green rattlesnakes. So Mojave rattlesnakes have an especially bad reputation among rattlesnakes because throughout most of their range, they are, well, we thought throughout most of their range, we thought in the US, they're neurotoxic, which means that if you're bitten by a Mojave rattlesnake in most places, bad news. It's going to be trouble. Not going to experience a lot of tissue damage. It might not hurt very much, but you could, like your chance of death is much higher. And so because of that, there's again, this big reputation of Mojave rattlesnakes for being bad news. But it turns out there's this funny thing about Mojave rattlesnakes. There is this chunk of their range, which is between Phoenix and Tucson in Arizona. And on this map, you can actually see it here. It's these blue dots. And in this geographic area, most of the Mojave rattlesnakes actually have hemotoxic venom. These are the same species. This is the exact same species. And for a very long time, because we didn't have great sampling, we thought that this was like the story with Mojave rattlesnakes, that throughout most of their range, they are neurotoxic. And then in just this one spot, they were hemotoxic. Well, it turns out that that's not the case because some researchers in Mexico started doing some work and looking at Mojave rattlesnakes in their neck of the woods. And they found out that, hey, all the rattlesnakes near us, they're hemotoxic. And once we got a more range-wide sampling of their venoms across their actual entire distribution, it turns out that there is, it just kind of goes back and forth. So in some places you have Mojave rattlesnakes that are neurotoxic. In other places you have Mojave rattlesnakes that are hemotoxic. And one of the things that's especially interesting about this, and the reason that this little phylogeny is on this figure, is that in fact, this venom variation is not explained by phylogenetic or population structure. So what that means is that across the range of this species, there seems to be interbreeding among these different snakes. And even though we know that venom is genetically related, we see that there are these very local patches of specific venom types. That's kind of interesting. It's not something we fully understand. But there is one other thing I want to quickly point out because this is really interesting and someone asked about it earlier. We have cases where we have these neurotoxic snakes. They've been shown in red. I pointed out a couple places where we have hemotoxic snakes that have been shown in blue. Anyone with really keen eyes, and I know it's hard to see up here, might have noticed a few of these little purple dots. They're in there and there and there. Would anyone like to guess what the purple dots are? Sam.
Both hemotoxin and neurotoxin.
Both. So there are some snakes that have both, but they don't occur as frequently. Kind of very strange. Yes, Chase.
So it's not like, it's not like a dominant recessive thing, like.
We don't know. We kind of don't know. We're going to talk about this more tomorrow, or not tomorrow, but Wednesday in our next class. But right now, it's just important to know that we have this variation, this key axis of variation. We don't know the actual inheritance pattern like dominance recessive, but we do have a clue about what's going on here. But I'm really excited to tell that to you guys on Wednesday. So that is something I want to put out there, and then hopefully no one wants to skip class on Wednesday. And if you do, now I hope you feel bad. Yes, SK. Yeah, in specifically in Mojave rattlesnakes, that is what they call it. They called it type A and type B. This was some researchers in the 1950s they decided, oh, this is type A and type B. Now, one of the things I will mention, just as like a note, this axis of functional variation, where you have hemotoxicity versus neurotoxicity, has been called type 1, type 2 by a really famous venom researcher, because it's generally characteristic of rattlesnakes. We tend to see snakes having one or the other. We don't see that many of these like middle guys where they're doing both. Yes.
I'm not sure if this is like a real question, but I've heard often that neurotoxicity is like worse.
Yeah.
Is that correct in saying that, or?
It is more toxic. That's the best way of saying it. So if I don't want to use a word like best or worst, because for instance, if I have a very low pain tolerance, then maybe it's not worse. Maybe I would rather be in the hospital on a ventilator versus being in screaming agony. But in most cases, probability of death is lower. That's what people mean, really. Okay, so we've got all this example of variation in snake venoms. Why? What's the point? What's driving this? Well, one of the important things that we know about snakes and venom is that they evolve in what's called an arms race. So this is a key idea behind predator-prey interactions, which is that your diet or the prey species, they exert selection on the trait. Because if you are a population of squirrels and you're being eaten by a lot of rattlesnakes, you've got to do something to stop it, to increase your fitness. And to do that, they often evolve resistance. This has actually been really well studied in one species of rattlesnake, this is the western rattlesnake, and ground squirrels that occupy the same habitats. And these ground squirrels are resistant to the venoms. So an adult ground squirrel can actually take a direct hit of venom, and in some cases, it'll just survive. And so there's kind of this back and forth between them, and it's called an arms race because it's likened to the Cold War, where if I'm a snake, maybe I've got a venom, it's kind of toxic, and then as a prey species, I become resistant to that. So then what happens to the snake? Well, I've got to have a more toxic venom, and so they develop a new toxin, or they evolve a new toxin, or they become more toxic. And then the prey has to respond again, and they develop more resistance. It kind of goes back and forth. So this is a really important ecological concept that is really well shown in venom. And another important thing to note is that we have a name for this type of evolution occurring between these two groups. This is called coevolution. So we can define coevolution as reciprocal evolution between ecologically interacting species. You might call it non-independent evolution between ecologically interacting species. But this is the idea that their evolutionary histories are intrinsically tied to one another because of some interaction that they're having. And then another thing that's really important to know about this is that these are typically considered to be asymmetrical relationships. And what that means is that the cost of a snake not having a good enough venom might be lower than the cost to a squirrel that is not resistant enough. Because if I'm a snake and I strike a rattle or a ground squirrel and it gets away, I probably still can try another day. And maybe I try and find a smaller squirrel, something like that. But if you're a squirrel and you are killed by a rattlesnake, you are dead and your fitness is 0. So that's it. This is called the life dinner principle, or that's a name given to this relationship. It describes the asymmetry between these interacting forces. Right. And so another thing that we note about venoms is that they act differentially across different prey species. So for instance, if you're eating a mouse, a lizard, seagull, or a bird, each of these 3 organisms has a slightly different physiological system. And so the same venom is not going to be equally functional, or the same toxin is not going to be equally functional across all 3. Yes.
Would the arms race be skewed since the snakes are eating less frequently and the prey have different predators?
That is an excellent question. That is such a great question. And it is not clear, or at least I don't have a lot of the primary literature to answer that off the top of my head. But that is a really great question, because you're absolutely right. That could be the case. Yeah, really great question. Yes.
Also about the
Mhm.
because generally you would think that the prey species, there's a bigger risk for them
Mhm.
than the predators.
Yep.
Does that affect the rate that they evolve defensively?
That is the hypothesis. So that's actually that's exactly what the hypothesis is that we should expect that selection is stronger for the prey to develop resistance than it would be for the snakes to have like more toxic venom. So we expect hypothetically the squirrels should be ahead in the race, but we don't know if that's actually true. We don't have great empirical evidence for it. And we've got some reasons to suspect that maybe that's not actually the case. So talking about these guys, though, again, the thing to remember is that if you're eating, these different taxa, they're going to be affected by venom differently. And there are actually lots of cool instances where this is played out in a biologically meaningful way. So we have examples of species with venom that actually have very specific functions. So for instance, this is a cat-eyed snake. These guys are bird eaters. Oops, sorry about that, you guys. I can't change it. So these guys are bird eaters, though, and I'll explain what's going on. You'll get to see it again in a second. And in fact, these guys have these toxins in their venom that are very toxic to birds and almost harmless to mammals, which is really wild. And a similar thing occurs in another species. So this is a South American species, and these guys have 2 toxins in their venom. They have more than 2 toxins, but there are 2 that we're referring to here. One of those toxins, high toxicity against mice, and it has almost no toxicity against lizards. And the other, high toxicity against lizards, almost no toxicity against mice. Yes, Chase.
Is it mice or mammals?
It's actually mammals, but I mean, these are tested in mice. So
Well, yeah.
here kind of interchangeable,
Yeah.
but yeah, it is mammal and lizard mostly, is what we're actually talking about.
So they
so-
would have they would harm a human. Or is it-
Hypothetically, but we're a little size plays a role.
Yeah.
Yeah. Yes, Sydney.
I'm not sure if this is a stupid question, but like, why are species so specifically adapted to one thing? Because wouldn't that make sense for it to interact with
That is a really great question. So it comes back to the idea of having a trade-off. So one of the things that we know about venom, I've got a great example about this that I'm going to share on Wednesday, is that it is costly. It is not a cheap thing. This actually goes back to the idea, and I think it might have been you, Sydney, who asked, I said, venoms don't arise spontaneously. And you said, do you have an example of why? Or do you have an example of one that has? And I don't. And the reason is because there's a great case, again, that I'm going to show on Wednesday, where the selection pressure to maintain venom has been lost. And in fact, in that instance, the venom has been lost as well. Yeah, because without selection to maintain it, you're basically wasting a bunch of energy. And so that's why there's a trade-off that comes with having a broad venom versus a very specialized venom. Okay, but so we've now got lots and lots of evidence, lots of really cool evidence about why we have snake venom variation, about the role that diet plays, and that is actually going to lead us right into talking about this article that you all read. And so what we'd like to do next, I'm going to turn it over to Dr. Gangloff, is to let you guys have a chance to look over your peer review assignments or your primary lit assignments. Yeah, absolutely, that works.
Let's take 5 now. We've got, what's it, 5, let's take till 3 o'clock even, and then we'll come back. When you come back, if you have a laptop and can get your file open with your summary and the other questions about this, that would be helpful, and we'll get started in a short workshop with that for class. So take 5.
I'll pause the recording here.
Perfect, thank you. All right, so we had our primary literature assignment number 1 is due today. And as you saw in your assignment, there's a couple steps as everyone submitted, well, hopefully everyone. Most of you submitted a link for the first part of the assignment. And then we said in the assignment, we're going to do a workshop with it. And then you're going to resubmit a revision of that. So today we're going to spend a little bit of time at least getting the workshop part started. But I want to walk you through some important information. The first thing you need to do is you need to share your document. So many of you have submitted a link to the document in the Google form as requested, but you didn't set up the document to be shared with other people. If you go into Google Docs and at the top right, there's a blue button that says share. You can just set it up to share with everybody in the Ohio Wesleyan community is the easiest thing because you'll be sharing it with each other as well. So please make sure your document is shared with the Ohio Wesleyan community. And then whoever you send the link to can access that document and make comments. Sam. You can if you want. That'll open it up to more people outside of the community. That some rando outside of OWU gets the link, they can edit your document. The chances of that actually being a problem are basically 0, but yeah, you could. If you share with the OWU community, that means that whoever's looking at it has to be logged in with their OWU account. Chris, go ahead. What's that? No, it should be. Yeah, you don't even need to do that. You don't even actually need to type that. Just get out of that menu and at the bottom, it'll automatically be there. See at the bottom? Yep. Boom. Yes, McKenna.
I was gonna say, if anybody's worried about it, you can set it so that people with the link can only comment on it so that they can.
Yes, thank you. I'll just repeat that to make sure everyone heard it. So McKenna pointed out that you can set it up so that other people with the link can comment on your document, but they can't actually go in and edit it. And that's actually probably what we're going to ask you to do is leave comments and not edit anyway. So that would actually work out fine. And then you're the only one that can actually make those changes. Actually, that's key. Thanks for pointing that out. So what I'd like you to do, make sure your document is set up to share. And what you're going to do is you're going to give and receive feedback from 2 peers in the form of comments on the document. So we'll spend a little time today getting started. You're going to share that link with 2 other people in class. You're going to get 2 other people's links, and then you're going to leave comments on their document. My only request on this is that you don't do this with people that you were in your small group with when you develop the criteria for what makes a good summary. The reason for that is I want people who have different criteria, I want their crosstalk between those groups. Because if you only talk to the people that were in your group, then you're all going to have the same criteria for what makes a good summary, and you're not going to kind of expand that thinking at all. So your assignment is to give feedback to 2 people and receive feedback from 2 people. Now, when you do that, you'll leave comments, and I'm going to say explicitly via comments. So I don't want you going in and editing anyone's document, just make comments about it. And then I'll be able to go in and see those comments and see who left feedback on the different documents. So part of the assignment here for the remaining 4 points is getting comments from someone, making revisions, and then giving those comments to someone. So I got a couple questions. Let me go through a few more things. Let me get the questions. I might answer your questions. If I'm psychic enough, I'm going to channel that energy. Then you'll make those revisions and then you'll have that updated in 1 week. So by next Monday, you'll have received feedback from other people in the class and you'll make updates to your document. Now you don't need to do anything new by next Monday. Because I already have the link for all those documents, right? And so you're just changing that same document. So you don't need to upload anything else. All you need to do is have it done by Monday. Couple other reminders with that. One is your summary. And I'm looking for feedback, giving and receiving feedback on specifically on the summary. Sorry, so I was better prepared to have a slide for this, but just on the summary part, not on that beginning part with all the questions. If you want to look through those, that's fine. What I'm really interested in you developing is that last part of the assignment, which is that summary in the format that we saw from the Herpetological Review current research summaries. As a reminder, those summaries are available in Blackboard as with all your readings. There's a PDF that has those readings that you did. So those are in Blackboard if you need to reference those. As you make revisions based on other people's comments by next week, you can also go back and make sure you answered all those first questions right. Like make sure that you read the assignment carefully and you're writing things in full sentences and answering all those questions thoroughly. This just gives you a chance to double check your work before you turn it in. I skimmed a few of the assignments that came in right before class and I noticed a few discrepancies there. So I just want to offer a reminder that this is your chance to make sure you've got all the assignment criteria set and everything like that. Of course, if you have any questions, you know where to go. Princeton. Is that okay? Sure. Ethan, go ahead. Yeah.
So as you're gonna be looking at seeing what comments are left, should we make sure not to resolve comments?
Oh, I didn't think about resolving the comments. How do we want to do that actually? Make sure I can tell who gave comments. So you can either leave the comments or you can make a note at the end. I received feedback from Mason and Michaela. And that way I'll know. So just indicate to some way who got feedback for you.
I think if you use the comment function on Google Drive, it should show the account next to it.
It does unless the editor then goes in and resolves those comments and they disappear.
Oh, okay.
But I think I can then go to comment history and pull those up too. I think you can. Can you do that? They won't disappear forever, those being comment history. Yes, you can resolve them. I'll just go to comment history and look. I love comment history. It's like my favorite. Athena. I did. Yes, I knew I could get 1 out of 2. Okay, so I don't have a slide. I'm recording this on the video, making sure that it's visible here. So again, the objective is to give and receive feedback on the summary portion from 2 people, and then make revisions based on that for a week from today. Sound good? Why don't we take, we don't have a ton of time, but why don't we take maybe just 10 minutes, get that set up, figure out which 2 people you're going to review. I mean, the easiest thing is just to swap with a couple other people. Trade links to your Google document via email or whatever, and get that set up, okay? All right, go for it.
I'll pause. Okay, so if I could get everyone's attention back, we're going to jump into the second half. We're going to talk about we talked about now venom, what it is. We talked about variation in venom. We've talked a little bit about one of the key drivers of venom variation or key selective pressures affecting venom variation, which is diet. You guys are gonna or have learned a little bit about that through your primary lit assignment, and you're going to give each other some feedback on that, which is great. What I want to talk about now for the rest of my lecture time is some of the diversity that we see of venomous herps. And so one of the first things I want to talk about is the venom delivery system. So this is how venoms are made. So the idea here, remember I mentioned that each of these organisms by and large is making its own venom. And it's going to be made in a specialized gland. So here in this rattlesnake diagram, we've got this big old gland right here. That's going to make the venom. It's going to store the venom. And then when it's in use, it's got some sort of mechanism of travel to be injected or to be, traumatically introduced to the host. So here it's going to go through this venom duct and then through the fang and into the injection site. Now, the reason I bring this up is because we actually see quite a bit of variation and venom and fang morphology across our major our venomous herp lineages. And so we're going to chat about some of those. I can click to the next slide, maybe. There we go. Looks like we got a little lag going on. So the first one I want to talk about is our big venomous lizard lineage. So this is going to be down here. So this phylogeny is basically the basic relationships of the snakes. It's not in great detail, but the outgroup that we're using here, remember that snakes are monophyletic within lizards. We have this lineage here that includes iguanas, chameleons, varanid lizards, and within that lineage is the Heloderma. So the Heloderma, these are the beaded lizards. In the US, we have one species called the Gila monster. If you've been out west, maybe you've seen it. Yes, Sydney? 2 slides, right? To the venom duct one? Yeah. Everyone good? Cool. So Heloderma is our only like very well-known venomous lizard. Again, the Gila monster is the only one that's native to the US. Now these guys have what's called a low pressure venom delivery system. And what that means is that for them to inject venom, they have to chew. So we saw earlier in that video, that rattlesnake, it bit the mouse, it was super fast, bam, hit it, and that was it. And in that moment, it injected actually a lot of venom. That's because it has, it's called a high pressure venom delivery system. But these guys, they actually have a venom gland that's in their lower jaw, differs from snakes, and they have these grooved teeth. So here's an example. These are some gnarly Gila monster teeth, big honking things. And if you look carefully, you can actually see this little groove on each tooth. And underneath these, like right underneath this jaw is where that venom gland is going to sit. And so while it's biting something and it's chewing, that gland is going to produce a venom. And through capillary action, it's going to rise up to the tooth and get introduced to the bite source. So that's characteristic of Heloderma. Yep, go ahead. I believe we're actually at 4, depending on whose taxonomy you use. The big ones are the Gila monster and the Mexican beaded lizard, but I believe they've also been split into the Guatemalan beaded lizard, and I think the 4th is like Heloderma alcatraz or something like that. Yeah. Okay, so moving on, our first big lineage of venomous snakes, the vipers. Yes, Sydney. Has to chew. So it still could be used for prey capture. I don't know as much about Heloderma venom as I do about snakes, and I've read that it's primarily for defense, but I've not seen a clear citation of where that was from. It could be used in digestion. So for instance, it might be that it's not as big a deal to incapacitate prey, but maybe it's helping digest. So that's not entirely clear to me. So I don't want to give you a definitive answer. But just because you're using a low pressure venom system doesn't mean that you can't have a very effective venom. We're going to see an example of that later, actually. But so the vipers, these are obviously a major radiation of venomous snakes. I'm sure that we're all familiar with vipers, even if we don't know it. One of the things to know about vipers is that they have a specialized enlarged fang called a selenoglyphus fang, or we call the fangs selenoglyphus. Now, selenoglyph fangs, there's a couple things we know about them. 1, they're very large, and 2, they're mobile. And so, for instance, you can actually see an example of this that I made up here. And if you can't see it well right now, you can come up to see it while we're doing the activity. I've got two sides of this little 3D skull model here that I should be more careful with. On this side, I've got an example of what the fang would look like when the mouth is closed. So the fang is actually folded up to the roof of the mouth, and it's just going to sit kind of back. And then when the snake goes for a strike, it actually swings that fang forward, and then it can be injected. So we can see an example of this. Sorry for the quality here. It's not a great video, but you can see that snake yawning just like before, stretching out those fangs. And one of the things I like about this little video is it shows you actually a couple things. One, those fangs can move independently. So just like your hands or arms, these snakes can move each fang independently. They actually have quite a bit of mobility. It's not just like, you know, your elbow has more mobility than just a single swing. And they can stretch their jaws really wide, and that allows them to have these very, very large fangs. One of the other things we know about vipers is that they have a very large venom gland, and it typically sits right behind their eye. So you often hear vipers described as having this, you know, big triangular head shape that is very characteristic of them. I'll give you guys, yes, Sydney.
So I remember when we first started taking this class, my friend and I were researching like venom snakes, and I forget the name of the snake, but there was one that had fangs that were like ridiculously long.
The gaboon viper has the longest fangs of any snake at 3 inches.
Yeah, so but like, why does something need that big of fangs?
Yeah, no, that's a great question. And it's not entirely clear, but one of the leading hypotheses is that it has to do with that your venom is more effective if you can inject it more deeply. And so, for instance, a snake like a gaboon viper, it's actually a very big snake. It's short, but it's huge. I mean, they have a head like that. And so that's the kind of snake that can eat something like a rabbit, very honestly. And there are other snakes that can eat rabbits too. If you think about the size of a rabbit, to really get a good injection, I mean, you need to penetrate there pretty deep. So you might need to have long fangs to do that. One of the things we know about vipers, or we hypothesize is true about vipers, is that mammals specifically were a big part of their diet. And so that's part of maybe why they have these longer fangs, is there's less of a possibility of them breaking or something like that. Yes, Princeton.
So when you say the fangs sort of like swing open when they shred, is that a function of them just like opening their mouths out
Nope.
at high speed?
Nope.
It's like some muscles.
Yep, they have muscles. They are specifically articulated with muscles. So for instance, I'm sure we've all probably seen shows, maybe you haven't, where someone like milks a snake and they take it behind the head and they do the thing. So it's actually even a little more dangerous than it looks, because they're really flexible, and they can poke fangs out the side and like swing them towards the back. They can't quite, but they can get pretty darn close. So they're more mobile than you might think. Yes, Sydney.
You were talking about how they have to stretch their fangs out.
Mhm.
Does that mean that it's pretty easy for them to like the correct word is slow to step?
They don't really dislocate them, but these are all, I mean, it is muscle attachment. So it is easy for them to move them. But that said, they do actually lose fangs too. And then they have replacement fangs that are right behind it, ready to come up like a shark's tooth. Well, let's look. This guy, for instance, which is just a 3D printed model, but you can see 3 replacement fangs that are all ready to come forward if they lose the one. And they do lose them regularly, just like naturally too. So, and actually, if you get a really good look at some of these, you can actually even see multiple fangs sometimes that are all kind of ready. Okay, a couple other things about the Viperidae. We have 3 big subfamilies in Viperidae. We have the Viperinae. These are going to be things like gaboon vipers. These actually include the Echis, which you read about in your primary lit article. We have Azimiopinae, which is a really weird viper. These guys live in China and East Asia, and they are very strange. They are like fossorial, which means they live underground. They're not quite monotypic because there's more than one species, but I think that there are currently 2 described species, both quite similar. They They're just kind of a goofy looking snake. And then our last group is the Crotalinae. These are the pit vipers, so named because they have these very large heat sensing pits. You can see it right in front of the eye right there. And those actually integrate with the ocular system. So it really does have like, give these guys some kind of heat sensitive vision, basically, like heat sensing goggles. Now, one of the other things that we know is that, in fact, all of the vipers that you see in the New World, which is North America, South America, Central America, they're all pit vipers. And that actually occurred because of a single invasion into the New World. You don't need to know that, but it's kind of a cool thing, I think. Yes, Sydney. Uh-huh. Gaboon vipers, yeah. Zimiopinae? That's this guy right here. So the family is the Zimiopinae, the genus is Azimiops, and I think that the common name is phase viper or fees viper because its scientific name is Azimiops fee. So in the US, we have 3 genera. I think that you guys have been introduced to these, so I don't want to spend too much time on it. We have Echistrodon, we have Crotalus, which are most of our rattlesnakes. And then we have Cistrus, our pygmy rattlesnakes, which includes Cistrus catenatus, which is Ohio's endangered pygmy rattlesnake. All right, our next family that I want to get to is the Elapidae. This is the other big major, like the one that everyone knows about as being another group of very deadly snakes. This family includes the cobras, the kraits, the coral snakes, and the sea snakes. The sea snakes, of course, being a major radiation because there are actually many species of sea snake. And then a number of other kind of small fossorial snakes that we actually see in the Pacific. Now, these guys have a very different fang structure than the Viperidae. So this skull here is our elapid skull. Again, I highly encourage everyone to come take a look at these if you get a chance, but touch them, be very gentle because they are kind of fragile. So these guys have kind of a very short fang, short fixed fang. So these guys can't move their fang forward and back like we saw with the vipers, and that actually limits how long they can get. And so, for instance, you really have to work to see the fangs on these guys. See that here. And in the US, we have 2 genera of elapid snakes, and there are only 3 species total. So we have 2 Micrurus. These are 2 coral snake species, the eastern coral snake and the Texas coral snake, Micrurus fulvius, Micrurus tenor. Then we have Microides, which is the, I don't know their common name, to be honest. Can't remember it. But these guys are kind of funny. They're really tiny. They're very small, like itty bitty little things. And they're very fragile. It's the only way I can describe them. Totally unlike any other snake that I've ever interacted with. They're very, very odd. And these guys actually are like insect specialists. That's what they want to eat. They kind of crawl around looking for worms and maybe little blind snakes, things like that. Actually, probably more blind snakes than insects. So those are our elapids. And then the last snake family that we're going to talk about are our Colubridae. So these guys are humorously, I call them. So this first off, this is called, I'm calling it Colubridae sensu lato, which means in the broad sense in Latin. It's a biology term. And the reason I'm using that is because historically these guys were called the colubrid snakes, but now the taxonomy is, well, it's in constant rearrangement as we learn more about their evolutionary histories. And so this very large group has actually been broken up into a number of smaller families to better describe the diversity within it. Yes, Sydney. Yes. Do you want the 3 species or do you want before that? Okay, here. Yep. Kill the fellow, red to black, venom black. Only in the US. If you go to Mexico, there are other species of coral snake that actually have the reverse pattern. If you go to South America, there are species of coral snake that have different banding, like just red and black. There's no yellow at all. And that, of course, always assumes that you're not looking at an aberrant coral snake that, for instance, could be melanistic and is jet black. Those happen too. But by and large, yeah, it actually is true. One of the things I always look for talking about coral snakes is a black head. Most of the other non-venomous species actually don't have a black head. They'll have like a red nose or like a white head or red head. The black head tends to be specific to the coral snake. But again, better to be safe than sorry. Yes, Sydney. Mhm. Yep.
Are they? What's the point of having short fangs, I guess?
Well, so the fangs are just as effective at delivering venom, right? And if anything, I mean, we don't know that there is necessarily a disadvantage or advantage. It's not something that's well studied right now. I think there was actually a paper that came out just maybe a year ago that suggested that a lot of the difference in fang morphology has to do with what you're eating and prey and how robust you need your fangs to be. So for instance, a short fang is probably less likely to break than a long fang, which might make them better able to eat reptiles and things that have tougher hides than mammals. Yep. Mhm. They do a little bit on the lower jaw. They have like a little pocket that, that they're not stabbing themselves with their own venom. But that is actually something worth noting. And I should have mentioned this, I forgot. Snakes have very good control of their venom. So not all snake bites are actually envenomations. So snake venom, as I mentioned before, is very costly. It's expensive. Snakes don't want to waste it. And if they can avoid using it, they will. So it's actually, I think it's like 30% of snake bites on humans are actually what are called dry bites, which means the snake bites, the fangs penetrate, but they choose not to inject any venom. And so the snakes could be the same here. So even if they poke themselves, for instance, with their own fangs, they just wouldn't inject venom. Yes.
How do they choose to do that?
I mean, I guess I don't want to necessarily use this metaphor, but I guess I will. You can choose when you want to or don't want to use the bathroom, right? So it's the same idea. They have muscular control over it, and it is like they have muscles that wrap around it. They can choose not to contract them, and those don't, eject the venom. I had a question over here. Yes.
Is there a great variation in general venom toxicity between electrolyte and
Yes.
okay. Because I was gonna say, if you've got shorter fangs, if you've got a higher toxicity venom. So
Yes. Yeah, there's a number of reasons. And actually that is another sort of key area. I didn't get deep into that when I was talking about the whole venom variation, but that's absolutely true. That just like at a very deep level, there are key differences that occur between the Elapidae venoms and the Viperidae venoms. But there are kind of multiple things could be going on there, because in part it could be that it's a function of what toxins they first recruited that started evolving in those lineages. And so those are more likely to stay. They're not going to, like try to recruit other ones. But that's something we'll kind of touch on more on Wednesday. So, yes, Sydney.
Just a follow-up.
Uh-huh.
Why, what makes the snake not?
It's a snake choice. It's a trade-off. Yeah, it could be. It could be if the prey is bigger, they'll inject more. It could be if it's a predator, how likely do I think it is that it's going to run away if I just bite it versus if I actually waste some of my venom. Yeah.
So we'll get into reptile cognition later. They absolutely can make
decisions based on complex sets of information.
Yep.
And they vary among individuals.
Yep.
Absolutely.
Okay, back to the colubrids, these goofy guys. So first thing about colubrids, these are, again, colubrids sensu latu. They are the majority of snake species, 63%. And there is a ton within this group, a ton of variation in fang morphology. So much I can't even cover it all, because you have some lineages that have well-developed rear fangs, some lineages that do not have rear fangs, some that have a very well-developed gland, some that do not have a very well-developed gland. One thing about all of these guys is they have a rear fang instead of a front fang like we see with these guys, or with the other two families. So I've got this is actually a hognose snake skull. If anyone happens to have a hognose as a pet, that's cool. Also know that they've got these big old spikes in the back. In fact, I think I've got a picture of a nice hognose showing off the spikes. So here you can see, and I've extracted venom from some colubrid snakes, and hognose snakes have enormous fangs for colubrids. They're really comparatively quite large. These fangs, unlike our vipers and elapids, they are not an enclosed hypodermic needle. They have a groove just like our heliderms. And similarly, their gland is not well encased in like a big sheath of muscle, so it can't produce that high pressure injection. So these guys also have to chew when they're trying to inject venom. And these guys are considered, they're pretty, mostly considered harmless to people, but there are certainly some exceptions. And I want to give one really famous example. This is the boom slang. This information is not as much of a thing you need to keep notes on, but it's really cool. So these guys, they have really gnarly rear fangs. These are an African species. Their venom, their toxicity is at the same level of toxicity, at least to lab mice, as like a Western diamondback rattlesnake. So pretty toxic venom. And there is an incredibly famous herpetology story about a boomslang bite because it involved this gentleman. His name is Carl P. Schmidt. He was a herpetologist in Chicago. And he received a juvenile boomslang from the zoo, and they wanted it ID'd. And there's some mythology around this, so can't say exactly what is true and what is not. But in some cases, they say he wasn't sure what it was. In most cases, and I think what's more reliable, he knew what the boomslang was, and he knew it was venomous, but he didn't realize how venomous it was. And so at some point during his evaluation of this snake, it poked a single fang into his thumb, put the snake back, and he then began to take notes on his symptoms, and he ultimately died. And so he took notes throughout the course of the 24 hours from when he was bitten almost up until the time of his death. And they're like extraordinarily detailed notes. So pretty fascinating story. Anytime you talk about colubrid venoms, you got to talk about Carl P. Schmidt. Yes, McKenna.
Don't they have human toxicity?
They do. That's exactly right. Yeah. So these guys actually, so not to get into the gory details, but these guys do disrupt blood coagulation. And in fact, your blood becomes very thin and it starts to leak out of cavities and things like that. So it's like it would leak out of your eyes and other orifices. Yes.
Are we going to talk about how anti-venom is being?
We can. We can do that. We can talk about a little anti-venom. Yes, Chloe.
Was there not anti-venom at the time of this, or did he just choose not to?
So there was probably not, and this is 1950, and certainly he probably did not have access to it because he was in Chicago, and again, it is an African snake. I know supposedly one of the things that they say is he refused medical attention because in one story I read, he refused it because it would disrupt his notes. It would like then he would no longer be a good case study because he's been treated. I don't think that that's true. I think he actually ultimately probably thought he was going to get better, just didn't realize how bad it was going to be. But I know that there is anti-venom for boomslangs now. Okay, one last snake group I want to talk very quickly about because they are super cool and goofy and strange. These guys are called the Atractaspis. This is a genus, Atractaspis. The common names are the stiletto snakes, burrowing asps. These guys are a lineage within Colubridae. their placement, I think, is pretty well placed now as being a true colubrid lineage. At certain points, they were thought of as being sister to the Elapidae. Don't think that they are now. But these guys have fangs, and they are super duper long. They're little vampires, and they poke out below the from the upper jaw. And the reason that they have it is that these guys are burrowing snakes, and they'll basically burrow into like a little mouse nest, and they will just like shake their head around and stab their prey. So in fact, they don't bite. They just stick like a fang out the side, and they will poke a little mouse and inject venom, and that's that. These guys you can't handle them. So like, again, I talked earlier, you might have seen people that have picked up like a viper and you have it by the back of the head, and they said it's really dangerous because they can get those fangs really close. There's no way to hold these snakes where you cannot be bitten. Yeah, so these guys are kind of a cool little lineage. Super flexible fangs. And then the very last couple of things I want to just very briefly touch on, because this whole lecture has just not shown any love to amphibians, because I know all about snake venom and I know a tiny bit about heloderm venom. But what about the other side of the herpetology spectrum? Well, we've got a couple of cases of potentially venomous amphibians. So there are two species of frogs. These guys were noted in 2015 as possibly being venomous, because even though they have a skin secretion, they have a skull that has these bony spines on it. You can kind of see, you can't really see them well here, but you can kind of see them if you zoom in. This is like a cross section of the tissue. And these bony spines poke up almost to the edge of the skin, and there is a gland that's associated with it. And in fact, these can hit predators with the spines and inject some kind of toxic substance. And it doesn't seem as though that substance is an alkali like we've seen in poison dart frogs. It seems like it's something the frogs are making. So by our technical definition, these guys are venomous frogs. And there are two species that I've listed up there. I don't know their common names. And then the other one, the last diversity thing, because I think this is so cool. Yes, Will. Yeah. Yes.
While we're back, I just want to
Mhm.
ask about an earlier slide.
Sure.
With the hog nose.
Yep.
That was an example of a taxa-specific venom, right, for amphibians?
I don't know enough off the cuff of their venom.
Because I've heard before that if you do get injected, because it's made for amphibians, it's more like a bee sting than an actual.
I think that there is a lot of individual variation with how people react to their venom.
I know you can be allergic to it too.
Yeah. And so some people have like really severe reactions where they get like a rash or like a welt. Others have no reaction. And that's actually, I think, it's probably a mix of host variations of like what would be my immune system reacting in the snake's venom to. I don't know much about their venom. I'd have to do some Googling and looking at the primary lit, and I haven't. Okay, so Sicilians, as it turns out, these guys are a little worm-like, kind of look like giant worms. But they're not, they're amphibians. They have actually really gnarly dentition, these teeth, if you haven't seen them. And just very recently, a study looked at these guys and said, it turns out that some of these guys have these glands right near their teeth. These are secretory glands, and they secrete enzymes. And guess what? Some of these enzymes, these are a lot like some of the snake enzymes that we see in snake venom. So maybe these Sicilians should be considered venomous. So this is a pretty recent study, and there's a lot going on here, and actually some of the things we're going to talk about on Wednesday kind of play into whether or not we might think about the Sicilians as venomous. But I just thought it was really cool, and it was something I really wanted to share with you guys because it was so neat. Case of a really potentially venomous amphibian. And with that, I'm going to close off for the day, and I will answer questions. But instead of answering questions right now, because we're running out of time, I want to actually mention one other quick thing, which is that on Monday, you guys will have your next quiz. And it's going to cover reptile diversity, reptile and amphibian diversity.
Actually, I'm gonna push it back a day.
Okay.
to Wednesday, the 23rd.
Okay.
I was just looking at the schedule while you were doing the lecture.
Okay.
And will include details on it for your class program for next month. But just to get on your calendar, it's actually Wednesday, the 23rd. That way we'll have time to finish up the venom and everything
Yep.
like that. And then I'll include details of that.
All right, so disregard this then. So with the remaining time, what I would like from you guys is to work on this activity. So this is something that's always really bothered me. I'm not a huge comic book buff, but I do enjoy Marvel films and I enjoy Spider-Man and things like that. And it's always really irritated me that this is the only character named Venom. And his powers have nothing at all to do with being venomous. Don't know who he is? Don't worry about it. It's not important. That always really bugged me. And since we were talking about venomous diversity, I thought this might be a really good opportunity to have some satisfaction in my life by asking you guys to fill the void in my soul where I'm missing some venomous superheroes or villains. So what I would like from you guys, what I'd like to ask you all to do is to work in groups, probably groups of 3 or maybe 4. And each of you all is going to come up with your own venomous herp themed superhero or villain. So you guys have the rest of the time to get started on this. And on Wednesday, I'd like for each of your groups to present a very short, probably 2 slides, maybe 3, PowerPoint on the character that you come up with. And so you're probably wondering, well, what do we have to do for this? Well, here are the requirements. These are also available online. You guys have the assignments is on Blackboard, so you can see them. And I don't think it's too bad. Basically, I'm asking each of your groups to come up with a venomous herp that you think is really cool and use something about them, their natural history, to inspire a hero or villain or anti-hero or anything in between. Just want a character who has superpowers based off of venomous herp and a weakness based off of venomous herp, and to provide us all with a little bit of background. We would like you to, in your presentation, first tell us which herp you chose. So for instance, on Wednesday, I might get up and say, I came up with the Western Diamondback Rattlesnake woman. That's her super persona. I don't know what her real name is. Probably Carol A. Troxa, which is very clever if you know the Western Diamondback's scientific name. And I would say this species is in the Southwest. They have a hemotoxic venom. And they live in very arid climates and they prey on a mix of lizards and mammals. And maybe I would say her superpower is she can make a very loud warning sound and she has heat sensitive vision. And she's got this weakness that she really struggles against heroes that are Neotoma, which are these big mice themed, or villains that are those mice themed. So, and we have a couple of other things, all that are available up here. So at this point, you guys have the rest of the time, break into groups, start picking your organisms, start working on your heroes. And next week, we will plan to see what you come up with. Dr. Gingloff, anything else?
We can go and check the two groups. And then
Yeah. So everyone's attention really quick for just a second. So after talking to Dr. Gingloff right now, and I agree with this choice, because I ran long talking a little bit about all the cool venomous herp diversity, we're actually going to do the presentations for these on Monday. So get into your groups now and start deciding what venomous herp you want. And then on Wednesday, you'll get some time to work on this as well in class. So it's not all, something outside of class.
So let's do this just to keep things paced for assignments. I know we're kind of mixing it up a little bit here. And I want to be clear. How about this? How about before class on Wednesday, you give that feedback to 2 other people's summaries? That's a good assignment to do between now and Wednesday. And Wednesday in class, you'll have time to work on the venom assignment. And then between Wednesday and next week, Monday, you can polish up your summary based on feedback you got from your peers.